Hello, ladies and gentlemen. I'm Daniel C. Montano, Chief Executive Officer of Angiogenesis Medicine. I'd like to thank you for joining us in this webinar presentation of Parkinson's disease as therapeutic angiogenesis, a treatment or even a possible cure. I want to, first of all, let you all know that our outreach program, and that is trying to identify people who could benefit from our medicines. We are very proud to announce that in the last 60 days, we have added an additional 20,000 people to our subscribers list. So we now have over 60,000 people around the world who are following us and participating in getting our monthly updates. Now I will make a comment that if you're new and you haven't signed up for our monthly updates, I would recommend strongly that you go to the website, wgm.care, and register with us to participate and get the monthly updates. Now, the last 30 days, we have outreached and attempted to identify people who are in the rehabilitation area, people who help people rehabilitate. It is our opinion that the activities that we're doing in the brain, whether it's Parkinson's, Alzheimer's, enhanced stroke recovery, it is our theory that we can revitalize those areas of the brain by adding perfusion, allowing those neurons to become functioning again. But even in that capacity, we still need to have a situation where people will be rehabilitated afterwards. So a lot of the questions that come in from us constantly are people that say, I have a problem with walking, I have a problem with speech. Will your medicine cure that? If we're correct, our medicine will allow the neurons that control that function to become normal again. And people will still need to be taught again how to make those neurons work with the rest of the brain to take care of those functions. So we believe there's a two-step program. The first step is to make the neurons functional again. And then the second part is to rehabilitate people. So I want to thank the hundreds of rehabilitation people who have signed up and joining and have sent us questions and had ideas, because we believe that there is a massive business, if we're correct, in helping the people that our medicine has now put them back on the point of being able to go back to a normal life, but they still have to be trained. Now, one of the other things that I want to emphasize, and that is that Jicha has a portfolio of 19 medical indications we're advancing. We're advancing the medical indication for growing blood vessels in the heart, where we've done 4 different human clinical trials with 100% success and 0 adverse events. And that is to address the biggest medical need in the world, stopping people from dying from heart disease, which is the #1 cause of death in the world. We have phase 2 data on our drug, healing diabetic foot ulcers. And chronic diabetic foot ulcers are responsible for 100,000 deaths in this country from people having their feet amputated because of gangrene, because these ulcers would not close, to the 30 million diabetics which exist in the United States and Canada. Our ability to heal these wounds, once again, in phase 2 trials, 100% success, 0 adverse events. Equally so, we have had tremendous success in FDA clinical trials with venous ulcers. Now, those are the things that are known to us. We have seen the data, we've been to clinical trials, we've seen the human outcome, we're highly confident we will succeed in those. The brain is a new area. The brain is a new area, and we're going into that excuse me, we're going into that with animal data that we're comfortable with, but we have to find out in the brain. Now, Dr. Jacobs and my wife Vika have been working extensively on the question of multiple sclerosis, Alzheimer's, etc. One of the things that's come up, which I'm very excited about, is in doing this process, they've been noticing that endothelial cell dysfunction appears to be a major contributing factor to all of these diseases. So we believe that insufficient blood flow to the substantia nigra area and the neurons that produce dopamine is a problem. But why is the blood flow disrupted? We don't know. So there's two steps to the process. Jack is going to talk in his presentation about this endothelia cell dysfunction. I think it's the most exciting part of his presentation. And for those of you who have seen Jack's presentations on Parkinson's before, please pay attention to this because I think this is a huge mind and thought opener. Dr. Jacobs is going to give his presentation, and I'm going to introduce Dr. Jacobs. Dr. Jacobs is a graduate PhD from Washington University, St. Louis, WashU as it's well known. He worked at Hitachi Pharmaceutical. He worked at Merck, where Merck had the patent on this molecule for 2 decades. Jack has been with me now for 22 years. We have a great relationship. Jack's the smart one and I'm the cute one. I hope you got a few laughs out of that, folks. So what I'm going to do now is turn this over to Dr. Jack Jacobs, our partner. Jack, it's all yours.
Okay, thank you, Dan, for that nice introduction. Before I get into my presentation, I just want to take one slide to update where we're at with our clinical trials for Parkinson's disease. We are approved to begin in Mexico. The Mexican FDA gave us the green light. We have a neurologist down there as our principal investigator. We have a nice modern hospital, and we're ready to go once the pandemic eases up down in Mexico. In Estonia, we are before the Estonia equivalent of the FDA, and they're basically reviewing the same application we submitted to Mexico. And Estonia is part of the EU, so that would be a highly visible trial once we get going there. We also recently have talked to a neurologist in Morocco, and there's a potential fast-track clinical development program which is possible there, and I think Dan will probably talk about that during the question and answer period. As many of you know, we submitted our application to the US FDA. It was the first time FGF1 was being looked at for a brain disorder, and they asked for one additional animal toxicity trial, which those studies are underway at the moment. Something I'm very excited about, we've talked to researchers in Minnesota who have delivered growth factors such as FGF1 intranasally, and they just squirt it up the nose. It goes up the olfactory nerve and actually gets into the brain in fairly large quantities. So obviously we're very excited about the possibility of looking at this form of delivery, and we're going to be working with them to test FGF1 in animal models of Parkinson's disease using this intranasal delivery form. Okay, I'm going to start now. I'm going to talk a lot in this presentation about blood vessels, blood perfusion, growing new blood vessels. It's a process we use all the time. It's called angiogenesis. We have over 60,000 miles of blood vessels in our bodies, enough to wrap around the world almost 3 times. And many of you are familiar, if we have blockages in our coronary arteries, we can develop heart disease. We know if we develop clots in these arteries going to the brain, we can develop stroke. But actually, there are over 75 human diseases that are a result of impaired blood perfusion. And some of those are just being recognized more recently, including the neurodegenerative diseases such as Parkinson's disease and Alzheimer's. And I'll try to convince you today that blood perfusion is a very important initiating event in Parkinson's disease. Let's talk about angiogenesis, the growth of new blood vessels. We use it all the time. The skateboarder is falling off his skateboard and cut his elbow. So let's look how angiogenesis is going to help this young man heal this scrape. Let's say this is the elbow of that young man. This is where he's gotten a scrape. He has a scab. We look underneath that scab. And here's angiogenesis at work here. These are new blood vessels formed in response to that injury. It's stimulated by the drug we're developing, FGF1. I'll talk about that in a second. These new blood vessels regenerate skin, not only the skin cells, but hair follicles, sweat glands, whatever it takes to heal that area. And our drug, FGF1, is probably the most potent stimulator of angiogenesis in our body. Okay, here's FGF1. It's a protein like growth hormone or insulin. We make it in our bodies. I actually was involved in purifying this when I was at Washington University. We go to the slaughterhouses in St. Louis and bring back cow brains. That's where FGF1 was first purified. Let me show you how FGF1 works. It initiates this process of new blood vessel growth, angiogenesis. So when that skateboarder scraped his elbow, FGF1 was synthesized by his body, released, and migrated to the wounded area. It binds to its receptor. Here's in yellow, the FGF1 receptor. And that starts this explosion of kind of metabolic activity with these endothelial cells. They begin dividing. They begin migrating. They actually begin chewing a little hole in the existing blood vessel and pop out a new tube here. This is the beginning of a new blood vessel. It gets elongated into a capillary. And this gets repeated thousands of times along this vessel to generate angiogenesis, to bring better blood perfusion into that wounded area and to heal that wound. Now, as I mentioned, the receptor of FGF1 gets upregulated in wounded tissue. So normal tissue, not much is going on with FGF1 or its receptor. But when you get a wound or the tissue becomes damaged or ischemic, starving, you get this tremendous upregulation. Here's the FGF1 receptor upregulated. Normal tissue might have 10 to 100 of these receptors per cell. In wounded tissue, it gets up to about 10,000. And when FGF1 comes in here are the yellow balls. This can be either introduced as a drug, as we're doing, or naturally in the body. You saturate all those receptors and stimulate the formation of new blood vessels. So we're using FGF1, as Dan mentioned, in 19 different medical indications. I'm going to show you a little bit of data just so you can see in people how our drug can grow new blood vessels in the hearts of patients suffering from coronary artery disease. I'll then move into the brain, show some animal data growing new blood vessels in the brain, and then finish up talking about Parkinson's disease. Okay, there's FDA clinical trial data where FGF1 was injected directly into the hearts of patients who had severe coronary artery disease. They could not have a bypass or a stent. This was basically the only treatment available to them. I'm very familiar with this. I actually supervised this clinical trial. And rather than give you the results, let me show you a news clip. ABC News went to our Cincinnati clinical trial site and interviewed our cardiologist there, Dr. Lynn Wagner, and also some of the patients. And you can see, I won't talk through this short video, but you'll see Dr. Wagner pointing in the patient's heart where new blood vessels grew 3 months after we injected FGF1 into the hearts of these patients.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
Just want to mention that woman we saw, Constance Donnelly, came in basically as a cardiac cripple. She was in a wheelchair. We had 3 dosing groups in that trial, and she actually was in the lowest dosing group, but probably responded the best out of all the patients we treated. Clearly, her heart was primed to respond to FGF1, and we believe her receptors were probably greatly upregulated to respond in that manner. Okay, let's move into the brain. Your brain is incredibly vascularized, probably the most vascularized organ in your body. You have billions and billions of neurons supplied by billions of capillaries, bringing oxygen, glucose, removing metabolic waste products. So the brain needs a very good and well-functioning blood supply system to function properly. Now, if we look at where the blood supply in the brain kind of meets up with the neurons in the brain, that's called the neurovascular unit. We start here with larger arteries and get down to the smaller capillaries and arterioles. So here's a brain capillary, a healthy one, fully open, supplying blood to this adjacent neuron and some of the other cells in the brain. So basically every neuron in your brain has a dedicated blood supply system, this capillary. And one can imagine if this gets blocked or clogged by atherosclerosis, damaged by inflammation or whatever, these neurons are not going to be doing as well, and they become ischemic and they die. Not shown here, but there's also stem cells in our brains. I'll show you some work later on about neural stem cells, but they also need a good blood supply to stay active. I'm not going to show you some data where we injected FGF1 into the brains of animals and grew new blood vessels in their brains. This will be in a animal model of stroke. So in this model, just like in humans, we can tie off an artery in the rat's brain. We tied off the middle cerebral artery and gave the animal a stroke, as shown here. So here's a stroke area, has a volume, has basically dead neurons in the core region, and then these are neurons at risk. Unless they get a decent blood supply reestablished, they're also going to be become dysfunctional. So we can do this in an animal, and these are slices of the rat's brain. If you believe in reincarnation, you don't want to come back as a laboratory rat in your second life. Anyway, these are the slices through the brain. You can see in the untreated animals, this white area is the stroke volume. We treat these animals for 2 weeks with intravenously administered FGF1. You can see remarkably a really nice decrease in that stroke volume. This is compared to this. So this is healing. This is regrowing blood vessels, regrowing neurons. And we can look at the blood vessels that were grown in these animals. These are the capillaries of a normal rat brain, healthy, abundant. What's not shown here are the millions of neurons that are being fed by these capillaries. We tie off that artery in the brain, and 24 hours later, you can see this decimation of the vessels in that rat's brain. And the neurons obviously are not functioning as well either. We treat those animals for 14 days with FGF1. You can see regrowth of those blood vessels. Not quite back to normal, but certainly better in the untreated animals. So here are the untreated animals. The brain is desperate to grow blood vessels to resupply those damaged neurons. These vessels grow back in a disordered way. These can even hemorrhage and lead to bleeding and further damage. So this is what we're hoping to see in patients that we treat with FGF1, where there's damage to the blood vessels and also to the neurons in the brain. Okay, I want to move now to talk about Parkinson's disease. As many of you know, it's dysfunctional dopamine-producing neurons are affected in Parkinson's disease. These neurons are housed in a very tiny area of the brain, deep within our midbrain. It's called the substantia nigra, nigra meaning dark. You can see these dark dopamine neurons. And researchers have shown using MRI that blood flow in this region is severely restricted in patients with Parkinson's disease. Let me show you the underlying cause of Parkinson's. We only have about 500,000 dopamine-producing neurons in our brains. Out of the billions of neurons, only 500,000 are the dopamine neurons. And actually, as we age, healthy adults, there's lack of blood flow as we age, as I'll show you in a minute. And we actually lose dopamine neurons as part of healthy aging, but not nearly as much as someone with Parkinson's, where we see this inexorable decline down to where a patient loses about 60 to 80% of their dopamine neurons. The symptoms of Parkinson's disease, the tremors, the gait disturbances start to develop. So we're hoping in our clinical trials, which will be starting, to regenerate enough dopamine neurons to pull people out of this area where they have their symptoms. Okay, let me show you, this is not our data, but data from medical centers on blood perfusion defects in Parkinson's disease. This first study looked at blood flow right in that small area of the brain, so the substantia nigra region. They used functional MRI, which can look at regional cerebral blood flow. And in the study, looked at healthy 21-year-olds in the substantia nigra region, had 100% maximal blood flow. As I mentioned, as we age, there's less blood flow in our brain. So here's a healthy 65-year-old, about 25% down. But look at someone with Parkinson's disease, about a 50% reduction in blood flow in the area of the brain that houses the dopamine cells, the ones that become dysfunctional in Parkinson's. And obviously someone with a stroke in that area is going to have a dramatic decrease in blood flow. Now, it's also known that people with Parkinson's develop other symptoms besides the motor disturbances. In fact, one of the first symptoms is loss of smell in some Parkinson's patients. They also have memory. They can have dementia in some cases. And we can look at perfusion in those areas of the brain as well as in those deeper areas of the brain. Here's a woman with Parkinson's who has dementia. Again, using an MRI perfusion scan. Blue is normal blood perfusion. Yellow is reduced, and red is severely reduced blood flow. You can see this woman has severely reduced blood flow in those areas in her cortex which are associated with memory. And there's no reason not to believe if we flood this brain with FGF1, treating both the dopamine neurons, but hopefully also reestablishing blood flow to these areas as well. Okay, we looked, FGF1 has been looked at in 2 animal models of Parkinson's disease, a rat model, and then the gold standard, which is a primate model, a monkey model. So I'll briefly show you data in both of those models. FGF1 is introduced into rats or mice, and actually they're tested on this rotating bar here. Here are normal mice. You can see they're kind of up top of the bar. Here are the mice treated with FGF1. They're hanging on, a little bit more difficult than these guys. But the untreated animals, you barely, here's a tail of one of them. They've fallen off this rotating bar. So these types of tests can be done on these animals after they're treated with FGF1. Here's the motor score skills. These are normal mice, mice that are given Parkinson's. This is done with a toxin that's directly injected into their brains. They become all the tremors and gait disturbances. Here's those animals treated with FGF1. We can see restoration of some motor activity, not quite back to normal, but certainly much better than these animals. Very importantly, if we look in the brains of these animals, we can see this is a substantia nigra region. Very little regeneration of dopamine neurons. They're staining brown. But in the treated animals, we see a nice healthy restoration of dopamine producing neurons in the brains of these animals. Moving on to the gold standard, the primate model. This is a marmoset, again, can be given Parkinson's disease by injecting a toxin into the brain. This is the MPTP toxin. This is basically a herbicide, and actually a very tragic occurrence back in the 80s. This herbicide was sprayed on the Seattle region on a marijuana crop. Some teenagers broke in and actually smoked that marijuana and came down with irreversible Parkinson's disease. A real tragedy, but the same toxin is now used in these monkey models of Parkinson's disease. Injected directly into the brain. Over a period of months, these monkeys come down with all the classical symptoms of Parkinson's, the gait disturbances, the tremors. So right here, we begin treating this group of animals with FGF1 intravenously. These animals get a placebo dose. You can see over time, over months, these animals begin to recover motor activity. They're about 75% of normal at this point. Untreated animals are doing much, much worse. Now, we could have kept this experiment going. Perhaps these animals would have gotten better, but at this point, we sacrificed the monkeys, looked in their brains. We wanted to see if we're regenerating dopamine-producing neurons in the brains of these animals. And clearly, as this next result shows, we are. These are slices through the brains of the monkeys in the substantia nigra region. The dopamine neurons stain brown. You can see healthy regeneration of dopamine neurons versus very little in the untreated animals. So this is a real disease-modifying activity. We're attacking the root cause of this disease. We're regenerating dopamine neurons, and we believe this leads to the better motor skills in these animals. Now, one other thing we can look at in the brains of these animals, both the monkeys and humans with this disease accumulate a toxic aggregated protein, very similar to Alzheimer's, where the beta amyloid plaque is in their brains. Patients with Parkinson's develop aggregated synuclein, and that's shown here in the monkeys. Monkeys with Parkinson's get this toxic tangle of proteins right within the dopamine-producing neuron region. You can see when the animals are treated with FGF1, we see a significant reduction in this toxic tangle of proteins. Not quite back to normal, but certainly better than this. So we feel these two activities, this flushing away of these toxic proteins, regeneration of dopamine neurons leads to the better movement scores in these animals. And that's why we're so excited to get into humans to see if we can replicate what we saw in these monkeys. Okay, as I mentioned, we're set to go in Mexico. Here's the clinical trial design. 3 ascending doses of the drug will be studied. No placebo group. The FGF1 will be given intravenously through a central PICC line. So this will prevent us from sticking the patients every day multiple times to give FGF1 or remove blood for testing. We'll be looking at safety and effectiveness, and the patients will be followed out to a year. So this is what is called a proof of concept study. This is the first time FGF1 has been looked at in any brain disorder. And the main thing we're going to be looking at is it's safe. We know it can get across the blood-brain barrier in animal studies. A certain percentage gets across. So if FGF1 is safe, we can tweak it. We can tweak the study. We can give bigger doses, larger doses, give it for longer periods of time to try to tweak out some effectiveness. Okay, I want to show you, and with 3 recent studies that really support a vascular cause of different brain disorders and can be directly related, I think, to both Alzheimer's and Parkinson's. The first study I'm going to show you is going to be a time-lapse kind of video of showing minor strokes that occur in the brains of patients that go on to develop dementia. It's really a remarkable video. You're going to see kind of these minor, they're red eruptions in the brain, and they're actually caught in real time. People are actually having these mini strokes that are asymptomatic. The patients or the doctors do not know that they're having these, but they accumulate and lead to dementia in the patients. And the authors speculate this could be going on, and perhaps even in Parkinson's, where patients develop dementia. So let me show you that now. from Toronto, looking in the aging brain. A week-by-week time lapse. Showing the MRI scan on the left, healthy, and on the right, someone with this white matter disease. Okay, they're going to show you now evidence for these tiny little strokes. Tiny strokes could accumulate over many years and result in dementia. So now they're going to show you the results of a small clinical trial they did. So there's the white matter disease. This is causing the dementia in the patients. Using this MRI technique to look at strokes as they are occurring in real time. So going through the weeks, you'll see a little red, there it is, a tiny stroke erupting. There's another one there. Again, so these tiny strokes with no notable symptoms by the patient. Okay, they're going to zoom in on one of these strokes as it is occurring. You'll see this MRI picking up the red explosion there. There it is, the minor stroke kind of comes and disappears. The stroke has disappeared, but you can see on the right, the white matter abnormality is still there. So these unnoticed strokes are common and are likely a cause of age-related disease. And preventing these strokes may prevent white matter disease-related dementia. We also think it could be involved with Alzheimer's and Parkinson's, and FGF1 certainly could create new vasculature in these patients. Okay, this is a second study from the very prestigious Columbia University, why our brains perform less well as we age. Their study, I think, directly supports what we're hypothesizing in these disease states, that a lack of blood flow to the brain can initiate such diseases as Parkinson's. They're going to show you here a lack of blood flow as we age normally causes our brains not to function as well. We forget people's names, where we put our car keys. Let me give you the punchline of that study before I show you the short video. They're going to show you that as we get older, less blood flow to our brains affects these stem cells. These are the stem cells that differentiate into neurons and other supporting cells. All these cells are needed for a healthy brain. And this video shows that as we age, lack of blood flow affects these stem cells. So we could not agree more. We agree completely, actually, with that video. Lack of blood flow to the brain, shown by Columbia, results in the aging brain not performing as well. We think the same exact process is going on in diseases such as Parkinson's. Now, Dan mentioned endothelial cell dysfunction in the beginning, and new research indicates that vascular dysfunction can initiate multiple sclerosis. I wanted to show you some data which supports that. Basically, the hypothesis is we develop vascular leaks, and researchers have looked into lesions that develop in multiple sclerosis and have seen that the blood vessels, excuse me, in those lesions are leaky. So let's take a closer look at this. Here's endothelial cell dysfunction in capillaries from an animal model of multiple sclerosis. So here's normal endothelial cells. These line all of our blood vessels. So inside of all of our blood vessels are these tightly packed endothelial cells that basically form a barrier. But in multiple sclerosis, in this animal model, you can see these endothelial cells become dysfunctional. They become disordered, bumps here. They become leaky. And so the thought is that in these lesions, there's leakage out of the blood vessels of such things as immune cells and proteins that become toxic. That is shown here. Here's a healthy blood vessel, optimal blood flow, you get healthy neurons. But for some reason, it's not quite clear what causes MS. Genes, smoking, vitamin D has been implicated, pathogens. But those blood vessels become dysfunctional. The endothelial cells become leaky. You have reduced blood flow. And very importantly, you have immune cells, proteins, fibrinogen. These components should never be near a neuron. They should be within the blood vessel, but they leak out. The fibrinogen actually is involved in blood clotting. So it's a very sticky protein that actually coats the neurons, makes them look even more foreign, increases the immune attack on these neurons. So most people think that MS is an autoimmune disease, which it is, but it's preceded, we think, by this the endothelial cells becoming dysfunctional, leading to leakage. And this can be seen in an animal model of MS. If you give FGF1 to those animals, you can restore and heal the damaged endothelial cells. So the animals are given MS. You can see the damage of the endothelial cells. This is looking inside the blood vessels. Treating with FGF1 over a period of days, we restore those blood vessels. Here's healthy endothelial cells. These cells are not leaking. These animals are doing much better. So we believe endothelial cell dysfunction, along with growing new blood vessels, kind of a two-pronged attack in some of these neurodegenerative diseases. You can look at histology. These are, again, endothelial cells, dysfunctional, leaking. As we treat with FGF1, we reestablish very healthy endothelial cells inside the blood vessels of those animals. So we're very excited. This is work from a large number of labs around the world, actually. These are all brain disorders where a suspected microvascular component could be involved. And all of these diseases could be potentially treated, we think, with FGF1 to stimulate new blood vessel growth as well as heal endothelial cell dysfunction. We talked about Parkinson's disease. ALS, we've just received approval to treat patients with ALS in Mexico. We'll be starting that clinical trial once the pandemic eases. I showed you data on MS. I've given a webinar in the past on Alzheimer's, chronic stroke, traumatic brain injury, post-traumatic stress disorder. The veterans very interested in this study. MSA is kind of a very fastly progressing form of Parkinson's. Chronic depression, very interestingly, FGF1 is much depressed in people with chronic depression. Vascular dementia. CTE is this boxers and NFL football players after repeated concussions can come down with this neurodegenerative disease. Okay, so we're happy now to take any questions. And then Dan Montano will give some concluding remarks. So thank you for your attention.
Jack, thank you for that excellent presentation. And as you know, Jack, we've promised everybody will be done before the hour of 9 a.m. on the Pacific or noon on the East Coast. So we've only got about 10 minutes for questions and answers. And we had about 30 questions, Jack, before the presentation started. This is a very lively group.
Okay.
The most predominant question that came in about 20 times is, can FGF1 promote tumor growth?
Yeah, it's a very good question. And we're checking for that. We've looked in previous clinical trials and the heart trials very carefully for any increase in tumor markers after giving FGF1. We've never seen any hint of that. And in animal studies where the FDA makes us give FGF1 at tremendous doses to animals, these are the toxicity studies, we never saw any incidence of cancer, no initiation of cancer in those animals. So we feel pretty confident that FGF1 does not promote tumor growth. However, we would not let someone with an active cancer into the trial because we think that's probably not being wise. So we exclude anyone with active cancer from any of our clinical trials.
Next question, will this treatment improve the Parkinson symptoms or will it just stop it from progressing?
No, we actually hope to halt the progression and then improve their symptoms, as we saw in the monkeys, as monkeys improved in their symptoms. So we're looking to regenerate dopamine cells in patients and hopefully reverse their disease.
Jack, this is a question which I'm going to take a moment to read because it's a long one. Focus ultrasound technology has been successfully used as a delivery mechanism for some neurobiologic therapies to enable passage through the blood-brain barrier. Has JICA considered this as an option to deliver FGF1 to targeted areas of the brain for Parkinson's disease? If so, has there been any actual experience with it to determine its effectiveness as compared to other clinical delivery methods?
Well, that's an excellent suggestion. We have not looked at focused ultrasound and actually would be willing to consider it. We look for help from all our audience. So yeah, I'd like to learn more about that. If you want to email me directly, jackjacobs@zhmedicine.com. That's something certainly we would consider if it would help our efforts.
Jack, next question. How safe is the drug and are there any known side effects?
Good question. Again, so in our clinical trials, we've never seen a serious adverse event that we could ascribe to the drug. Of course, before we go to people, the FDA makes us inject this drug, at 10 to 100 times the dose that we're going to use in humans. So at those supraordinary doses, we do see, it's a potent growth factor. So things start to grow. The most sensitive organ is the thyroid. The thyroid starts to enlarge at 100 times the dose we're going to be using in our Parkinson's trial. And then if you stop the drug, the thyroid comes back to normal. So it's a reversible side effect. So no, we don't think we're going to be seeing any adverse events at the doses we're going to be testing in our Parkinson's disease trial.
Okay, the most common question we seem to have gotten, and let me answer it, is how do I sign up to participate in your clinical trials? Go to our website, zgm.care, and there you can fill out a form and that will get you listed. That will also get you signed up for the update. So once again, for a lot of these questions, how do I participate? How do I keep informed? Please go to zgm.care. Okay, Jack, another question for you. Will having DBS exclude me from future clinical trials?
I'm sorry, having what? Could you repeat that?
DBS, deep brain stimulation.
Oh. In our first clinical trial, yes, the FDA has asked us to exclude DBS, but just only in our first trial. I would hope in our phase 2 trial that we could enroll patients with DBS as well. So good question.
Jack, we have a question from a doctor in Morocco, our new friend. He
Good.
wanted to say, nice presentation. Thank you, Jack. What would you like to say to him?
Well, hope to visit Morocco someday, Marrakesh. Sounds like a very nice city. So thanks for viewing in.
Okay, next question, Jack. What is the difference between VEGF and FGF1?
Okay, yeah, VEGF is a angiogenesis growth factor. Also been looked at clinical trials. In the heart, it grew new capillaries, but unfortunately they became leaky, so that patients developed cardiac edema. That trial had to be stopped. But it is a potent angiogenesis growth factor, and actually FGF1, as part of its mechanism, stimulates the production of VEGF. So FGF1 is kind of above VEGF, and the two of them, I think, in concert, produce a robust angiogenesis reaction. So good question.
Jack, next question. Are Parkinson's and disease and Alzheimer's disease related? And does Parkinson's disease make Alzheimer's disease worse?
Yeah, it's more of a question for a neurologist. We believe both of these diseases are initiated by lack of blood flow to different areas of the brain. We know patients with Parkinson's can develop dementia. And I've heard of patients with both Parkinson's and Alzheimer's. And obviously, we're going to be treating patients in our clinical trials with just one or the other of the diseases at this point in time.
Jack, we have all kinds of questions on the criteria for the clinical trial, and that's going to need to be the last one. So why don't you address that in general?
Yes, in general, we have inclusion criteria, what you must have to get into the trial. Obviously, you must have Parkinson's disease. Exclusion criteria, if you have it, you can't be in the trial. For example, if you're pregnant. And we can, we'll be posting those on our website as we get going, and they can be looked at. So patients can go to zgm.care, sign up for our Parkinson's clinical trial, and then we'll be contacting them with the inclusion and exclusion criteria.
Now we're going to go to the conclusion, Jack. So would you be kind enough to put up my next slide?
Sure, here we go.
Ladies and gentlemen, Jicha is a Las Vegas-based biotech company. We are developing biological drugs which stimulate therapeutic angiogenesis. Our drugs address the leading causes of death in the world, lack of blood flow to a tissue or organ. I want to comment that our pipeline, once again, is 19 different medical indications, from heart disease, which you saw the ABC News clip, we grow blood vessels in the heart, to healing diabetic foot ulcers. And the brain is a new area for us. The brain is a journey of discovery. We have not been in the human brain before, and therefore the most important question is it safe in the brain? Our business is discovery. How does the drug work? What are the medical indications? How does it apply? And when is it safe to get the outcome? We are designing and carrying out clinical trials to demonstrate that the drug is safe and effective in humans with diseases that are caused by a lack of blood flow. And we interact with government regulatory authorities to obtain approval to sell the drug. Now, one thing I want to go into, one of the questions that came up was somebody asked the question, why are we doing clinical trials in Mexico, Estonia, Morocco? The answer is very simple. The United States FDA requested that we do another series of animal trials of our molecule in the brain because they wanted to see some more specific evidence that is very unique. We're doing that. We're proceeding. Now, the COVID-19 pandemic shutdown has deferred a lot of the activity, and that is being done, and we're going to get it done as soon as possible. In the meantime, we were informed that we could go and apply in Mexico. They would accept our animal data. They did. Estonia has said they will accept our animal data. We'll find out in a couple of weeks or months if they do. And in Morocco, we are now exploring with people there to initiate a clinical trial in Morocco. Why Mexico, Estonia, Morocco? Very simple. Time is important. If we dose people in Mexico and we find out that the molecule is safe at level 1, good. And we go to level 2, good. It's safe there. Then we go to level 3. If it's safe there, that is a level where Jack and I and the math indicates the molecules should sufficiently clear the blood-brain barrier to equal the level that the monkeys got. We want to know that outcome as soon as possible. If we discover it is not safe, why would we want to spend another $3-4 million in the United States to verify the fact it's not safe? We can stop at that time. We can be in a situation that if we see outcome and we say, well, do we dose 3 weeks longer? Do we take higher? What do we do? We can play around with it and learn quicker and faster. The most important thing with these diseases, whether it's Parkinson's or MS, is time. We don't have time to waste 5 years looking at our belly and wondering what's going to happen. So by going to Mexico, Estonia, and Morocco, we're going to be able to have data in humans on whether it's safe and whether it works before the animal studies will be done in the United States. And to the people out there that we promised that we would not fool around, we would get it done, we are pushing as hard as we can. Also, one other comment. All 3 trials that are budgeted in Mexico, Estonia, and Morocco, all 3 of them together come in at less than half of what we would have been paying in the US. Now, we're still going to do the US. Do not get me wrong. This is still the place that matters the most. The brain is a new area. The most important question in our journey of discovery is it safe in the brain? If, as in animals, we know where we can go to and it's safe, then we can experiment with dementia, Parkinson's, multiple sclerosis, endothelial cell dysfunction. We can experiment once we know it's safe, and I believe start a whole new discovery. Now, a lot of people have sent emails to us saying, oh, you don't have a finished product yet. Why are you doing this if you don't have a finished product? Well, ladies and gentlemen, this is a journey of discovery. Jack and I have been at this for over 20 years. We've gone through $140 million. A lot of that, my money. We are discovering things every week, every month. We are pushing the horizons. Some people want to be on that journey with us. Some people want to see if there's hope for their condition. Some people with Parkinson's want to learn what's the latest thing, what's going on. Is there hope I can be treated? And so when we uncovered the endothelial cell dysfunction in multiple sclerosis, the question was, does it apply to Parkinson's? Does it apply to Alzheimer's? We're finding out. Now, we have prepared white papers on many indications. So if you go to the website, zgm.care, you will see white papers on Parkinson's, white papers on Alzheimer's, white papers on multiple sclerosis. Also, if you go to YouTube and you go to Angiogenesis Medicine on YouTube, you will see our portfolio of prior presentations. This one will be edited and up within a week or so, but we are continually doing presentations. And some of you ask questions, is Parkinson's similar to Alzheimer's? Is multiple? Take a look at our data. We think they may be. We believe that the vascular dysfunction is reducing the blood flow to different areas of the brain. And what you see are the symptoms of that. So we are moving forward. We anticipate trying to get government approval as quick as possible. In the United States, we have a long path. We have been led to believe maybe we could have a quicker path in Mexico. Maybe we could have a quicker path in Estonia. Maybe we could have a quicker path in Morocco. We don't know, people. But I would rather have 4 horses in the race than none. If there is a delay in the United States for whatever reason, I still want the answers as soon as possible. Mexico is having a COVID-19 disaster, and the hospitals there are not doing clinical trials. Estonia shut down because of COVID-19, and now they're opening up. We are exploring in Morocco because it is an area with a lot of people. And the area there, and we're getting brilliant neurologists in all these locations who have different ideas. The neurologists in Mexico suggested, well, let's do this, let's do this. The doctors in Estonia said, well, maybe we can do a spinal tap and do this and this. Everybody, we're finding out, does it work? Is it safe? Time is the essence. We have a booklet we've prepared called treatment for heart disease, strokes, recovery, Parkinson's, etc., is therapeutic angiogenesis. If you would like a copy of this small booklet, it goes through the biology, how it works, what, etc., and gives basic data, and it lists the white papers and gives other presentations we've given. If you'd like this, please email me at dan@gchipmedicine.com. Here is the white paper on Parkinson's disease. Jack, as you can see, wrote this paper in January of 2020. The endothelial cell dysfunction is not in this paper. We're looking into that, and we're learning more every month. And I think that 60 days from now, we may have a few more discoveries, especially how FGF may be able to address the fundamental problem. And by the way, if we can keep you dying from heart disease, if we can keep you dying from diabetic foot ulcers, if we can help you recover from a stroke so you're back to normal, if we can help reverse your Parkinson's, your Alzheimer's, then you're going to live another 20, 30 years healthy, in my opinion. Now, if you want to follow us, that's your privilege. If you don't want to follow us, that's your privilege. But we invite all of you along for the journey, because I find it to be an exciting journey. Our next upcoming webinars in October will be mailed out to you soon. We're working on the ALS and the traumatic brain injury. I would also make a comment. We've been giving private webinars to groups on can therapeutic angiogenesis extend your life an additional 30 quality years. And we've had a tremendous response to that, because that sort of encompasses everything. I'm 72 years old. The fact that I may only have 30 or 40 years left upsets me. I'm looking for another 20 or 30 years on top of that. We want to make you aware of our science, and we want to develop networks of motivated and informed individuals. As I said earlier, we have over 60,000. We're reaching out every week, every month to tens of thousands of people all over the world. And I want to make sure you understand something. We get feedback from research scientists in Europe, from research scientists in Asia, who said, have you tried this function to look at the medicine? Have you tried this? Have you thought about intranasal? Have you thought of this? Have you done this? We are basically becoming a lightning rod for people who are saying, I understand the problem and I agree with you, but nobody has a methodology to treat this. FGF1 can treat it. Try this, try that. And we are learning more, and we usually get swamped after one of these presentations by medical scientists and research scientists who say, hey, I got a better idea. Have you tried this? And the good news is, people, Jack is a scientist. I'm a businessman. I spent a lifetime getting things done. I was an investment banker for 30 years. I started 100 companies. Nobody ever said I was pretty, but people will tell you I get it done. And we've committed to you that if this molecule can treat Parkinson's, we will get it done. Once again, email me at dan@gchipmedicine or visit our website, zgm.care. Ladies and gentlemen, we thank you for your time. We hope that we've contributed somewhat to your enlightenment on this topic. And please understand, we are committed, and we wish you all a good day. We pray that God blesses all of you. Thank you.